Hi everyone, my name is Aavani. I'm a PhD student at Texas Tech University, and today I'm going to talk about one of my research projects, pathogen evolution when transmission and virulence are stochastic, and it's supervised by my advisor, Sean Rice. There have been a lot of mathematical modelings on the host and pathogen interaction. Most models have been focused on understanding the condition for the population of the pathogen to invade and affect the population of the host as some major phenotype of the pathogen and the host. For example, two major phenotypes of the pathogens are transmission, which is the rate that the pathogen transmits from the current host to the future host, and virulence, which measures how much damage the pathogen can impose on the host. But pathogens, like their host, are biological organisms, and their phenotype can evolve. We have some pathogens that evolve to be less virulent, such as myxomovirus, which is causative agent of myxomatosis in rabbits. Experimental studies showed that when these pathogens first introduced to the population of the rabbit, the invasive population of the rabbit in Australia, the first generation of the pathogen was highly virulent, that could kill 99% of the infected individuals. But the later generation of the pathogen became less virulent. The possible explanation for this this evolution comes to the transmission process of this pathogen. Myxomovirus grows inside the host, and for its process of transmission, it needs an infected host to transmit to the future host. As the number of the hosts decline dramatically, there is a strong selection on the pathogen to be less virulent, so in other words, to harm the host less to maximize its transmission. On the other hand, we have some pathogens that evolve to be more virulent, such as the bacterium Vibrio cholerae, that is a causative agent of cholera in population of humans. Again, if we look at the transmission process of these pathogens, we see that bacterium Vibrio cholerae induce diarrhea on its host, which itself can harm the host. So therefore, increasing the transmission goes with increasing the virulence. There are a lot of mathematical modelings to study the evolution of the pathogen, but nearly all of those models are deterministic. What do I mean by that? It means that the major phenotypes of the pathogen, such as transmission and virulence, and the fitness of the pathogen, which is a function of the transmission and virulence, are all treated as fixed values. For example, let's look at this figure. Here we have a population, and each dot corresponds to the individual. And we see that on x-axis, we have expected offspring phenotype, one value for phenotype. And on the y-axis, we have expected fitness, one value for fitness. In other words, in deterministic models, what we're doing is working with one value for each individual. But in reality, we cannot exactly determine the transmission or virulence. Or in other words, the transmission is inherently stochastic. What do I mean by that? I mean that given that the pathogen infects the current host, we cannot exactly determine how many hosts will be infected by this pathogen or descendant of this pathogen. Also, virulence is inherently stochastic. Again, conditional to the infection of the current host, we cannot exactly how much damage the pathogen can impose on its host. Fitness is a function of the transmission and virulence, and fitness inherits this stochasticity. So in stochastic models, we treat transmission and virulence as random variable, and the fitness, which is a function of the transmission and virulence, also becomes a random variable. So let's come back to the figure. Here still we have the same individual, but instead of working with the expected phenotype or expected fitness, I work with the phenotype. But phenotype is a random variable, and it possesses a distribution of possible values. Fitness also has a distribution of possible values. But now what I see here is the joint distribution of the phenotype and the fitness. So to study a stochastic trait, to study evolution of the stochastic trait, what we're doing, we're working with a stochastic price equation. Here is a stochastic price equation. I read from the left. The first term is the change in expected value of the mean phenotype is equal to the summation of the three terms. The first term, it's a double square bracket, correspond to the first figure that I talk about. The covariance across the population between the expected value of the phenotype, phi naught hat, and expected value of a relative fitness. So if we see here, covariance across the population is positive. The second term that only happens when we treat the fitness and also the phenotype as stochastic or random variable, so is the probability covariance. Within each individual, we have a covariance. So if you see that each individual, we don't have expected value, and within each individual, we have a covariance. And in this hypothetical example, within each individual, we have a negative covariance. We have a bar sign, which means that we take the average over the entire population. And the third term captures any term that can happen during the process of transmission, such as mutation or recombination. Now, if you see the stochastic price equation, we have a relative fitness. And relative fitness is a ratio of the individual fitness to the mean population fitness. But I like to work with the absolute fitness. For this reason, we expand the relative fitness using a Taylor series, and so we have the summation of several terms. In this talk, I only focus on the effect of the first term. The effect of the other terms, we already also studied those, and if you see that there are effect of the variance of the transmission, variance of the fitness, and also covariance of the individual fitness and the mean population fitness, and they have their own effect. But now, for this talk, I'll only focus on the first term, which is the relative fitness is a ratio of expected value of the absolute fitness to expected value of the mean population fitness. Now I have my approximation. I substitute the expansion of the omega into my price equation, and here I have the equation for the evolution of the transmission, beta, and evolution of the virulence, and nu. Now, this is a summation of two types of covariance. Again, covariance across the population between the expected value of the transmission and fitness, virulence and fitness, and the covariance within each individual. So transmission and fitness, virulence and fitness, taking a mean across the population. Now, fitness, we know that is a function of the transmission and virulence. And depends on the biology of the host and pathogen, how they interact, we have different equations for the fitness. In this talk, I only focus on those type of pathogen that goes inside the host. And for the process of transmission, they need to infect the host and then transmit to the new host. And also there is a trade-off between the transmission and virulence, which means that so they cannot hurt the host most in order to maximize their transmission. The good model for this is a SIR model, simple SIR model. So the population of the host in a SIR model divided to three groups: susceptible, infected, and the recover. And with this assumption, I can find the fitness of the pathogen by finding the per capita growth rate of the infected individual. So if I find the per capita growth rate of infected individual, so I can approximate the fitness of the pathogen. So you see that this is a function of the transmission beta and virulence nu, and S is a susceptible. Now I substitute this back to my price equation. Now we have expansion, so as a summation of four terms. The first two terms in each equation, evolution of a transmission and virulence, are again the covariance across the population, covariance and variance across the population. They have already been studied in the deterministic models. But there are two other terms, the covariance and variance within each population, covariance and variance within each individual. These are the new terms, and these are as important as the corresponding terms in the deterministic models. For example, we see that the covariance between the transmission and virulence can be negative, such as myxoma virus, which means that increasing the transmission goes with decreasing the virulence. It can be positive, for example, such as Bacterium vibrio cholerae. Increasing the transmission goes with increasing the virulence. In other words, the biology, different biology of the pathogen can change the sign of this probability covariance, the sign of the within individual covariance. So I introduced a theoretical term, so a new theoretical term that can only happen when we treat the phenotype as a stochastic. So it's called probability covariance. But the question here is, can we measure it? Experimentally, can we measure it? So it's hard. If we think about this, this captures different things that can happen to each individual and is different from other individuals. But technically, if we use the clone of the host and clone of the pathogen, and we infect the clone of the host with clone of the pathogen and drive the transmission and virulence, so therefore, we probably have a distribution that we can approximate the probability covariance. No experimental study has ever been done to just drive the probability covariance, but we could find an experimental study by Bell et al. In 2011 that they used the clone of the host, Daphnia magna, and clone of the pathogen, Pasteurella ramosa, to find the covariance between the transmission potential of the pathogen, which is a good approximation for transmission, and mortality rate of the host, which is a good approximation for virulence, in two environments, high food environment and the low food environment. But the only problem that is not clearly capture our probability covariance, here they use the different strain of the pathogen to increase their data set. But still, it's a good approximation for what we call probability covariance. This is a very, very interesting experiment because it shows that when we change the environment of the host in the high food, the probability covariance between the transmission and virulence is negative. When we change the environment now in the low food, the probability covariance between the transmission and virulence is positive. So which means that not only the sign of the probability covariance between the transmission and virulence is dependent on the biology of the pathogen. It also dependent on the biology of the host. So different environment can change the physiology of the host that can affect the sign of this probability covariance. So therefore, we expect to see in the high food environment, there is a negative contribution to the evolution of the virulence. And in the low food environment, there is a positive contribution to the evolution of the environment. So what I did here in this talk, in summary, so I introduced a new type of covariance, new type of correlation. So it's called the within individual correlation, but we call it probability covariance. And I argued that, so this probability covariance can be estimated using the clone of the host and clone of the pathogen, and also the sign of the probability covariance is not fixed. It can be changed due to the biology of the host and biology of the pathogen. That's my talk. I can take a question. Yes?
So would these be correlated environmental effects in sort of standard quantitative genetics terms?
It can be, yes. We, in our original research, we talked about this, the first term, so it can just capture the standard quantitative genetic effect. And also, I know that Troy Day and Sylvain Gandon also work on the deterministic effect of this, and also they had the same, similar results.